Hello viewers, I'm Adway Zacharias from Dr. Chowdhury's research group, and I'm going to be presenting my work, which is deciphering metal-protein interactions by mass spectrometry-based proteomics methods. So the reason why we're interested in doing this project is because metal cofactors have been involved with directing cellular function and metabolism, and the interaction between metals and proteins have been implicated in a variety of diseases such as cancer and neurodegeneration. So we decided to use our expertise in mass spectrometry and couple it with the already well-established technique of immobilized metal affinity chromatography to develop a novel methodology to screen proteins for their affinity towards metals. Now, the affinity towards metals has been documented in cases such as zinc, iron, nickel, and many more, but we want to direct our primary focus towards zinc. So how do we go about building a column? So if you look at the first figure, you have a Sephiros matrix that constitutes the bulk of the column, within which the zinc is immobilized. And the zinc is immobilized by means of NTA. Once we introduce our sample into the column, certain proteins that have an affinity towards zinc will bind to the column, while the other proteins are simply washed out. Figure 3 explains how the experiment is conducted. So we add what's called an equilibration buffer to prepare our column for the protein. We then add the sample and then centrifuge the column. During this step, the proteins that remain on the column bind to zinc, and the proteins that don't bind to zinc are simply washed out. Now, the proteins that are bound to zinc need to be removed from the column, and this is accomplished by using EDTA or by using a pH gradient. Once the proteins are eluted, they can be then analyzed using mass spectrometry. We have results to show that our column works well, and we've used peptides and proteins that have documented good interaction with zinc, such as oxytocin, bovine serum albumin, and carbonic anhydrase. If you look at figure number 4, we have a mass spec of oxytocin that's been eluted from the column after having been bound to zinc. We've also tested the column at the protein level by using proteins such as BSA and carbonic anhydrase. And if you look at figure number 2, we have an STS page of BSA and carbonic anhydrase that have been identified individually as well as in a mixture. So we actually spiked BSA and carbonic anhydrase together, introduced it into the column, and we found that the column was able to isolate both of them together. The ultimate aim of this project is to eventually use proteins from an entire cell lysate and screen all of those proteins for potential binding activity to zinc. If you look at the highlights section, this is exactly what I'm working on at the moment. We're also interested to try and see if proteins that have not previously been documented to bind to zinc are identified. And if they're identified, we would like to model their binding sites and also try and see what level of specificity it has towards zinc. We also are interested in carrying out mutations to see the effect of what mutations have on the affinity of the protein towards zinc.